be talking about proteins. Proteins are part of many structural components in a cell or an organism. They are enzymes that are used in chemical reactions, and they're very important in terms of the actual function of a cell. They are also part of muscle fibers, and they play a role in contraction and or moving things around within animal cells in particular. The monomer of a protein is an amino acid. An amino acid is made of, it has carbon, it has an amine group, and it has a carboxyl group, and it has R groups. Now there's 20 different types of amino acids, and what makes the amino acids different are their different R groups. Now looking at this overhead here, you can see here's an example of some of the different types. The part in white is the part that is representing the R groups. You can see the R groups are different for each one of these. Some of the amino acids are polar, some of them are actually charged, that is, they might be positive or negatively charged, and some of them are completely non-polar. So you have some that would repel each other in the structure because they are not dissolving in water, and they are also repelling against each other. So those are the different R groups. Now these are going to be put together into polymers of these amino acids, put together into what we call a polypeptide. Sorry about the moving around. Now a polypeptide is just putting many of these amino acids together, and it is going to be put together again through dehydration synthesis. So here's an example of each of these. So it's just representing, it has R as the R group. So again, these are just different ways of writing that amino acid. So you can write it this way or this way, okay? And if you were to put them together, what's going to happen is a molecule of water is removed. So from the carboxyl of one end, I'm going to remove that hydroxyl. And I'm going to take one of the hydrogens off of there, removing the molecule of water, and forming the bond then right here between the two. In other words, the hydroxyl came off, and now that bond is happening with the nitrogen instead of it'll have one hydrogen hanging off instead of two. So this is the peptide bond. And over here, where would it be? It's going to be right there. Okay? So these then are forming chains of the polypeptides. Now, the proteins have various types of structure. The first type of structure is called a primary structure. The primary structure is simply just the long chains of amino acids. Now, at this point, it's not really what we call a functional protein. Instead, it's just got many peptide bonds. It's very long. Now let's say, for example, I have a positive and a negative amino acid here. So these are two perhaps charged ones, and maybe these are two nonpolar ones, so they're going to repel each other. The secondary structure will form because those parts that are attracted to each other will come together and form a bond, and the repelling parts will push these two apart. And so it will create a fold in the chain. Now remember, these chains are very, very long. So they could be, 20, 100 long or so. And they're going to fold, or sometimes they'll form pleated sheets. But basically they're going to interact because of the parts that are charged and or polar that are interacting with each other, or the parts that are repelling each other. All right, and then if you take that folded structure and you make the parts that were now far apart from each other can interact, then what's going to happen is a 3D structure called a glob. Nice scientific word for you there. This is a 3D, or what we call a functional protein, and most enzymes are in fact globular proteins. And the fourth type of structure is called a quaternary structure. This is where you have more than one glob that is connected to each other. All right, so I'm going to show you some overheads of these, because better than my actual drawings here. So this is just representing the primary. Whoa. Sorry, cat dog fight. This is representing the primary structure of an amino acid. So it's just, I'm sorry, of a polypeptide. So this is just many of the amino acids put together. Now these amino acids have names, of course, that you might recognize, maybe some of the things that you take as a supplement, like lysine or something like that. Then here's an example of the actual folding or pleating. So you can see it can form what we call a helix, which is kind of like wrapping around, or pleats, and so this would be secondary structure. The tertiary structure then is going to be taking these guys and making them interact and making the 3D type of molecule. All right, and then the quaternary structure is taking more than one of these structures and putting them together. In other words, like this is a glob and that's a glob and that's a glob. Hemoglobin is an example of a quaternary structure molecule. Now this is just showing you the actual types of, upside down, the types of interactions that can occur in the different parts of the chains based on the R group. You could have things that are pushing away from each other because they are nonpolar. You can have disulfide bridges because proteins, some of them contain sulfur. You can have ionic bonds, or you can have polar interactions forming hydrogen bonds. So those are the types of interactions.